Giving Tuesday live at BTI. We continue to move on throughout the Institute, hopping around at a feverish pace here. We are joined by Lucas Mueller and Susie from the Mueller Lab. Susie Strickler. And we're going to learn a little bit about what happens here with regards to bioinformatics and some of the other breeding research and the things that they're doing here. So Lucas, tell us a little bit about what your lab does.
Yeah, so our lab is a bioinformatics lab. That means that we are a so-called dry lab, not a wet lab. So we don't get as many fancy things that you will see in other labs like PCR machines and pipettes. But we have some really nice.
At least you stay clean, right?
We stay very clean and dry. So, but we have some fancy computers. They are downstairs, hidden away. And so our labs look more like, office space. But we do some interesting things, I think, nevertheless. So we work with bioinformatics, which means we analyze mainly genomic data, sequence data, some metabolite data, proteomics data, and so forth. And I guess the two main pillars in the lab are kind of genome sequencing and annotation, and the other pillar is creating databases for other researchers to access and ask questions of that data. And so the databases are basically, again, subdivided in kind of two different conceptual types. One is breeding databases that are mainly used by breeders in Africa right now. We have created databases for plants such as cassava and yam, sweet potato, and bananas. And the other type of database is more molecular biology-oriented databases, genomics-oriented databases for researchers. And there we have a database called SGN, the Solanaceae Genomics Network, which is a database for tomato and potato and all the different Solanaceae, petunia, peppers, eggplants. There's a lot of Solanaceae.
Yeah.
And the other one is citrus screening, and we'll talk to the people who create these databases as well. So, but first off, I think we can hand it over to Susie,
Okay.
and you can talk a little bit about what you're doing.
Absolutely, yeah. So I'm a research associate in Lucas's lab. I think I've been here about 6 years now. So one of my main projects here, I guess I fall into the genomics category of the two things that Lucas mentioned. And so I do more with genome sequences and then also looking at gene expression and some various things of that nature. The big project we're working on now is the coffee genome. So we've sequenced Arabica coffee. Arabica coffee is the one that people generally think tastes good. So that's usually used in drip and all your fancy espresso and that sort of coffee. We also sequence the two parents. So Arabica actually has a rather complicated genome that's derived from two parental species. So we sequenced both of those. And then the other thing I do here is, at this point, kind of people internationally, we have some collaborations with the Philippines as well for coconut and some other projects there. So there's a lot of different genomics projects going on that we're involved in. And the consulting's been great because I've been able to work with a number of people here at BTI. There's some good projects we've had with Greg Martin's group looking at genes that are involved in pathogen resistance in tomato. So that'll be a big thing that I'll be working on in the future. And also in the past, we've identified in projects that I've been involved in, looked at two genes that we know are involved in pathogen resistance. So it was great to be a part of that as well. So any questions about what we're doing?
Yeah, I have a question in that it seems that bioinformatics is becoming a pretty big, how necessary and important it is in research. Why is that exactly?
I think because the data sets that are being generated by people in the wet labs are becoming so large that you pretty much have to have these skills in order to be able to mine other ends and extract biologically relevant things from them.
Yeah, we can just sequence so easily today. You know, this was a huge headache how to get sequence data for example, the entire genome. But today we can sequence a genome in an afternoon, basically, almost. Well, I'm exaggerating.
We're going to hold you to that.
But it's really become so easy to get the data, and that has kind of shown that really the bottleneck now is actually analyzing the data and you know, having around that actually can do this kind of work. And so that's why bioinformatics has become so important. But in the future, I think what we'd like to do anyway at BTI is that you know, we have these courses for the BTI postdoctoral staff, so that everybody will learn about bioinformatics. And every scientist who does biological science will be at least part-time bioinformatician.
And so here in the Institute, you have, Susie, you have a weekly session, right? On bioinformatics. What goes into that?
Yeah, so every Wednesday we have it, we call it Bioinformatics Hour. So on Wednesday, and anyone from the Institute and also people from Cornell somewhat frequently come. And they can just come with any bioinformatics related questions. So if they have something like they're trying to identify a gene and they have some sequence data and they want to analyze that, or they have some transcriptome data and they want to get candidate genes based on that, things like phylogenetics, so looking at how different species are related to one another, anything and beyond, even those topics, they can come and ask us and sort of get some advice on how to process their data.
The way you can kind of, you know, most people think of data is how much can my phone hold? You know, like, how much data are we talking about here? And like,
well,
what kind of comparison would you?
We're talking about many terabytes.
Okay.
So that your phone cannot hold it. I think in about maybe 5 years it will.
I would say a typical genome project, all of the data combined takes at least a terabyte. All of the different file types that are involved in the genome assembly and then finding the genes in the genome.
So yeah, it's a lot of data. It's a lot of data.
And then there's the whole analyzing of that side. Is that done by the researchers, or is that part of what?
We do that as well. Well, it depends. Sometimes, so with the consulting, sometimes people want to learn how to do the analysis. In that case, they do it. You know, like I'll show them how to do it and I'll give them some assistance. But other times people are really busy in the lab and you know, they want us to do it. So, you know, either way, whatever people want, we try to work with them.
So I guess the people who are involved with creating of the breeding databases are over in the resource center. So should we move there, or how does this work?
Sure, we can move there.
We can move there?
Yeah,
we
let's
can
move.
Okay.
Thank you.
So let's go over there and briefly talk to these people.
Thank you, Susie.
You're welcome.
Garbage can. So you may have seen a story we put out about Lucas's work with cassava and some new breeding technology that will be used. So here's the team.
This is the team who works on the breeding databases. Like to start off, maybe Guillaume, you would like to start to say something about cassava work?
Our work here is based on developing databases to assist plant breeding. Plant breeding aims at improving crops, especially for our projects here. We work with crops in Africa, staple crops, and thus we aim at providing to the local institutes tools that are helping them to fasten and improve their selection cycles for those crops. In my case, I work mainly with cassava, which is a root, and we have here other people who are involved in different crops, which are also root or what we call tubers. And for those crops, we have key traits, which are usually tolerance to pests or diseases. Drought stress in Africa is also a very big topic. And we interact very closely with those institutes by working with people from those countries. So here with me, I have Uba from Nigeria, who is working at the National Institute, and Agwe also from Nigeria, working in an international institute. And they are coming here to visit us to get trainings with those databases and exchange information on those and the selection they make on that. I will.
Sure. So my name is Alex Obona, and I'm from Nigeria. I work specifically on YAM-based, but we also work as a team on all the databases. So YAM-based is actually developed for African YAM project, which is run out of IITA in Nigeria, and there are 4 countries and 7 breeding programs that are participating in this project. You have Nigeria, Ghana, Benin Republic, and the Ivory Coast. And the main objective of this project is to focus on genomic improvement of 2 species of yam that are based in West Africa, which is the Ascoria rotundata is native from West Africa, and then you have the water yam that is from the Asian countries, and then so they are working on improving this crop or this species, and their main objective is to strengthen the different breeding programs that are participating in this project. And basically, what we do is, along with developing these databases, like for example YAM-based, which we aim that it will be like a one-stop database for this crop, and again provide tools and functionalities that enables these breeders and breeding programs to carry out their routine breeding activities on a daily basis. Our passion is really to develop a very user-friendly database, and that is very intuitive. In that way, we can serve them better, and the objective of the project is achieved.
And my name is Brian. In addition to cassava, which Guillaume talked about, and yam, which Alice talked about, we have a few other crops, and I work mostly on our sweet potato database, which is in collaboration with something called the GT4SP project, which is bringing genomic tools to sweet potato breeding. The ideas are similar to what you've heard for yam and cassava. It's in collaboration with 2 main programs in Africa, in Uganda and Ghana, and a program here in the United States at North Carolina State University. And the idea is to improve yield, other agronomic traits, and improve vitamin A content, which can address important micronutrient deficiencies in sub-Saharan Africa.
Okay. All right. Thanks so much, everybody. Really impressive work.
Should we move on? We have another
Sure.
office where we can go to, and where people will... Thanks, guys, by the way. And we can talk to the people who create the citrusgreening.org website, which is a website about the citrus greening disease, which is devastating citrus plantations in Florida mainly. So these are the people.
Here they are.
Do you need a chair to sit down?
Yeah, maybe I'll sit over here.
So this is Surya with
wheels. Great.
.org project, and Prashant Osmani, who is also working on that project.
Great. How much time do we have?
We have about 6 minutes, five minutes. Let's say 5.
There's another office we go to after that, actually.
Oh, after this?
Yeah.
Okay.
So 3.
Okay.
3 minutes. Okay.
We'll keep this short. So we are trying to do this as a team, so not just one person. And so, okay, the main thing is that this is a dry lab. So we do only mathematics, specifically work on computers. If we had time, Lucas would have loved to take you down to the server room and show you where all the numbers are actually crunched. But in summary of what is done in what is citrus greening, first of all, it's a critical disease of citrus in the state of Florida. The losses right now since 2004 and data have been around $9 billion or so. 6,000 plus jobs have been lost. And this every single county, in every single citrus growing county in Florida has been infected as of today. So it's not a threat, it is a clear and present danger. The next big danger is in California, so those folks are really freaked out. The disease, and the government is throwing money at it. The growers are very scared. There are many growers who have been doing this for generations and they have gone bankrupt. So this is a very important problem. What we primarily...
Yeah, so what we are planning on doing is like to create an infrastructure which will help us to tackle this disease better. And as Lucas had probably told, like we have one like a genome and another is a breeding portion of our lab. We are focusing here on more on the genome side, where we are creating more infrastructure for genome. We are improving genome and also trying to get a more better understanding of the genome. So it's a big collaborative project. That's one of the beauty of this project. And
so yeah, so two points. When Prashant said that improving a genome, you may have heard around 2001 the human genome was published. Did you know that people are still working on it? They're still improving it. So genome is never finished. It always has to be improved. So the genes on the genome have to be improved too. The analogy.
When you say improve a genome, you mean improving the sequence.
Improving the sequence.
Not the actual gene. I think that's important.
We are not doing GMO.
Exactly.
Yeah, we are making it more accurate. When we say improving, we are making it more accurate. The other point is that this is a big collaborative project. So the way, if you think about when you are looking for information, you maybe go to Google and you search for it, right? Google has terabytes and petabytes and femtobytes of information in the back end. So we don't have data at that scale, but scientists who are working on this project have similar problem. They are generating massive data sets, which are gigabases and terabases of data, terabytes of data. And they need tools to look through it. You can only use Excel for a few things. Then you have to scale up. So that's where we come in. We build essentially data mining tools for people, for molecular biologists, for breeders who are working with these large data sets, and they need to understand how to target and to generate hypotheses, target specific genes of interest, and then perform experiments, because experiments are very expensive to perform. So they need a good hypothesis generation system before they go in and start spending thousands of dollars and man hours or woman hours on it. Yeah, that was, am I missing something?
I think that's pretty much it, yeah. And one of the other focus we are doing other than the citrus cleaning is working on the tomato genome, where we have a genome is basically a large amount of information, and to get more sensible data out of it, we have to get a better characterization of genome. That's what we call as an annotation of a genome. That's something we do it. We get this genome and analyze it on our massive servers, which takes even days to run it.
Months. Sometimes. I mean, when we say our servers, we are talking about machines with 1 terabyte of RAM, 64 CPUs. So that's probably more powerful than your typical laptop. So yeah, this is a lot of number crunching and data analysis, and also presentation. See, if you have a lot of numbers, you can generate a few summary figures, but you have to present it in a way in which people understand. So there is a whole life cycle of data analysis which has to happen before something useful comes out of raw data. So yeah, and big data and all that jazz. But it is true. Parts of it are true. So
yeah. Okay, thank you very much.
Yeah.
So maybe let's briefly go and see another researcher. So we have a lot of visitors in the lab, international visitors, as you've seen with the breeding project. But we have also other researchers who visit to analyze their data sets and learn new techniques. So we can talk to one of these visitors if you
Sure.
have time.
Yep. Got 3 minutes. And we want to thank Harmut Forrester for making a gift to our campaign here on Giving Tuesday.
You guys have time for a
From the Mueller
moment
Lab.
for a little interview, and this is all live. So, so maybe you just take it from here. So Rubab and Sean are visitors from Pakistan, and they are analyzing data sets here. So
So our major objective of the project is basically to do RNA sequencing of is susceptible, and basically we are focusing on cotton leaf curl disease so that we can find out some differential expression genes in the resistant line, and these differentially expressing genes basically will lead us to fetch all the important genes that ultimately can be used for breeding and genetic engineering purposes so that we can improve cotton just like our country in Pakistan. So BTI is one of the very nice institutes where we have find a chance to come here, and thanks to Dr. Lucas who has provided us a chance to be here and work with his genius and wonderful team. And Shani.
Yes, so me and Rob both are working on the same project. So ultimately our goal is to improve the cotton leaf curl disease, which is very devastating disease in Pakistan. So here we have found an opportunity for to work on the developing technique like RNA sequencing, so we can find some genes. So it's a great opportunity for students like us to work here and to do great things. Thank you.
Thanks so much.
Thank you so
Best
much.
of luck.
yeah.
Thank you.
So it really seems like your team has quite the international representation here. Is that just kind of go to show how important this work is to pretty much everyone in
I think
in
so.
the sciences?
Yeah, I think so. That is one point, but I don't think that we are much different from any other lab at BTI. I think attracted a lot of talent from everywhere in the world. So it's a very international environment. And it's very nice to work with all these people with different backgrounds.
It is. I think you can really feel the impact when you hear people talk about their, doing the work that they're hoping to kind of take home in a way.
Yeah, exactly. I think it's something really great that we're doing here at BTI.
But Lucas, thanks so much for your time.
Thank you.
And plenty more information on the Mueller Lab on our website, bti.cornell.edu. We're going to take about a 4-minute break here, and then we're going to hop up to the Schroeder Lab, where we're going to learn all about the important work up there being done on the roundworm. So thank you again for all those who are supporting us on the Giving Tuesday, and for everyone tuning in. We'll see you in just a few minutes.